Alright, today we're going to cover bacterial cell division and replication, and I'm going to do this on the board just because there's a lot of diagrams that are easier to explain when I actually draw them out. So you guys can follow along with the slides as I go. So I just drew this here, so we're going to start. So bacteria, as you can see on the slide, they divide asexually, which means that they just make a copy of themselves every time they divide. And that process is called binary fission. So you can see kind of the steps of that process here. So you have the bacterial chromosome. Remember, it has one piece of DNA that's circular, so we just call it one chromosome. Then what happens is that the cell will elongate, and it'll form this what's called a Z ring. So this protein called FTSZ, this protein will come into the middle of the cell and make this ring called the Z ring that'll start to separate the two cells. And from here to here, of course, you see that the chromosome has duplicated, so you have this process. Of course, where is it? Yeah. So of course we have replication, which is the DNA being copied. So you have two chromosomes, replication has occurred, the Z ring's forming. Now, and eventually what happens is that this Z ring gets smaller and smaller. So this is constricting inwards, and then you have this kind of break off here with the two cells, and it forms what's called a septum in between. The septum is just the new cell wall being formed between these two cells, so that eventually they can separate like that. And just some other small details. Let's see if I'm forgetting anything. Yeah, so FTSZ, this is a homolog. A homolog just means a protein that's very similar in structure and probably evolved from the same origin. So it's very similar to tubulin in eukaryotes. So if you remember, tubulin makes up the microtubules, right? Those long filament things in the cell that kind of pull apart the chromosomes. So it's a very similar protein in bacteria here as well. Let's see if I'm forgetting anything. Yeah, and then also, this is just a small detail, but this FTSZ protein, if we zoom in right here, we'll see that if we have, the membrane, we'll make this the cell wall. And you have the membrane, kind of have this structure. It's so let's say you have this FTSZ, and it's kind of anchored into the cell surface by FTSA and then ZIP A. These are just two proteins that basically anchor this Z ring into the cell surface so that the division can properly occur. So for this whole lecture, we're kind of going to look at E. coli as the model. So in E. coli, we have what's called the doubling time, or the time it takes for a cell to undergo this complete division. And that is, for E. coli, that's around, that's around 20 minutes. So doubling time, 20 minutes. So it's the cells are multiplying by 2 every 20 minutes. And this means that sometimes you might get, you have to do a calculation for how many cells there are at a certain time point. So let's say they tell you start with 100 cells, and then it's E. coli, so let's say the doubling time, 20-minute doubling time. And then let's say they say after 2 hours, how many cells will there be? 2 hours. So then of course, if you want to calculate how many cells there are at the end, basically, there's 6 20-minute increments, right? 6 20-minute increments in 20 hours. So that essentially we're the cells are dividing 6 times, so 6 generations, which means that what we'll do is we'll multiply 100 cells by 2 to the 6, because each time we divide, we multiply the number of cells by 2, and then we have 6 divisions. So then you just calculate, this would be, and then that's the number of cells you had at the end of that time period. So that is how you calculate that. Yes. Yep, okay, so we can move on to replication, which is the process that we'll be looking at in detail here. So this will get a little bit complex, so just make sure you stay with me here. So it's a very complex process that involves many different enzymes that have different roles. And there's kind of the same 3 steps that we've been seeing as in like transcription and translation, right? So first we have to initiate your replication. So first we have to initiate, so initiation. Then what we're gonna do is we're copying that DNA. So we're elongating the DNA strand, the copy, so elongation, right? And then of course, after we copy all of the DNA, we're gonna have to do termination here. So we're gonna have to stop that process and then split off the new DNA from the old DNA, essentially. So there's a general overview. Another kind of weird thing about this process is that, excuse me, in E. coli, replication actually takes 40 minutes. And like we'd say, right, but the doubling time is only 20 minutes. So how can replication take 40 minutes? So essentially the way they solve that problem is, let's say, so we have the chromosome, right, and it's being copied. So it's kind of, we'll do this like, we can do this. So let's say it's being copied, this inner thing will become the new, right, another circle will form here and then the 2 chromosomes will separate. So as this first one is being like copied, from the strand that's already being copied, you can have another division start to occur. So now we have, this is the original, we have this new one being formed. Within the new chromosome, we have another new chromosome slowly being formed. So it's like this replication is occurring, it doesn't wait until the original replication is done, it just keeps firing over and over. So you literally have like this, 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 this. You can think of it like that. So chromosome, new chromosome, off of that another new chromosome. So it's just like continuously happening. So that means that overall we can satisfy this requirement of the doubling time being 20 minutes because it's continuously dividing in that way. Yeah. Then we also have the idea of semi-conservative replication. So when we replicate, remember, we're replicating the DNA. So we have 2 strands. Let's say we have, I'll just draw them as like 2 rectangles here. So we have the double-stranded DNA that we start with. Now when we copy this, what's gonna happen is that we're gonna preserve one copy from the original, or not one copy, but one strand from the original DNA. And then we're gonna create a second strand here. So this orange is new, and then the white is old. So you can see we call it semi-conservative because when we have, after we divide, each new daughter cell has half of the DNA that the original mother cell had. So that's why it's, let's see this back here. Semi-half conservative. We're conserving half of the DNA, semi-conservative. Awesome. Okay, so let's actually, let's start getting into the actual process of replication itself. So we'll start with initiation, right? Because we need to actually start doing the process. So how that's gonna happen is that on every bacterial chromosome, we have this region, you have a single region called what we call ORI-C, or the origin of, and so we have this region of DNA called the origin of replication. So as you can expect, that's where the actual replication process is going to start. So let's look at what actually is contained within that sequence. So eventually the goal will be that at this site, right, if we have the double-stranded DNA, we're going to want to open it up here like this so that we can get some enzymes inside to start copying the DNA on both sides, right? So this origin of replication is going to need to be opened eventually. So let's look specifically at this region. So we're zooming in on the ORI-C. This whole thing. So you'll see that there's several different elements. First, we have what are called color these in with color. We have what are called DNA unwinding elements. These are on the slides, DUEs. So as you can expect, DUEs are places where this DNA is going to start to come apart really easily. So we have three of them in E. coli, and they're, I don't know if you have to know this, but so when I say BP, that means base pairs. So that many nucleotides long. So there's 13 like ATGC, there's 13 of them within each of these sequences within the OREC. And let's see. Yeah, and these DUEs, these DNA unwinding elements, they're full of A's and T's. They're mostly A's and T's out of the 13. And the reason why is because, I think we mentioned this earlier, but A and T have, so if we're bonding an A and T, and remember the bonds in between the two nucleotides are hydrogen bonds, right? And we compare it to G and C. Between A and T, you can form 2 hydrogen bonds, whereas between G and C, you can form 3. So that means the bonds in general in DNA between G and C are much stronger than the bonds between A and T. So essentially, we want a lot of A's and T's in these DNA unwinding elements, because this means the bonds are weak between the 2 strands, so they can be separated pretty easily, and we want to like separate the strands so that the enzymes will go in and then start copying the DNA, right? So that's how that works. On this OREC, we also have these regions, several regions, that bind what's called DNA A. DNA, should I write it down? Lowercase a. DNA A. So this DNA A is an enzyme that will specifically bind to these locations on the OREC. And the DNA A, essentially, it just helps these DUEs, these DNA unwinding elements open up. It facilitates that opening of the DNA. And then what it also does is it recruits DNA B. So this guy is like, DNA B is my friend, right? So he's gonna like bring his friend over. Now DNA B is another enzyme, but this one's specifically what we call a helicase enzyme, helicase. So when you think of helicase, you should think of unzipping. So helicases are enzymes that always literally come in and unzip the DNA, forcefully go in and then separate like this. So you can see DUEs with the weak bonds already starting to separate. This DNA A comes in, it fully separates these guys. This DNA B comes in and it completely opens up that DNA to make it available for copying now. Let's see if I got anything here. Yes. So essentially after we do this whole process, remember this is all initiation, all of this. After we do this, we're going to have this new open strand. We're going to have an open region at the OREC now. So essentially what our DNA looks like now after initiation is we have this open region like this. And I'll draw the chromosome with 2 strands of DNA. So we have this open region here. And we call this, let's see if I get to this here. Yeah, actually I'll get to that later. But essentially, let me zoom in on this portion here. So essentially what we have is you have this DNA still wound up, and then you have it open here, like this, if you zoom in here, right? And this is happening by the action of the DNA B helicase enzyme like this. So once this is separated, what's going to happen is that this DNA B helicase will move down, so move down here, and then keep opening up all the DNA as it's going. So you can imagine this guy is going to keep traveling this direction, and then eventually all the DNA here is going to get opened up as well, and that will traverse around the entire thing eventually. So, and then once we open up the strand like this, right, these, DNA usually likes to exist in a double-stranded format. So now we have these single-stranded DNA things, and that's like not It's very unstable, right? It's as the DNA wants to coil back up and then become this like double helix formation that we always see, right? Double helix, but it's being separated. So what we do is we put what are called single strand, right here, SSBs, single strand DNA binding proteins, or SSBs, that just come in, they just stick onto this DNA and make sure that it stays stable here. Also, we have, so this DNA molecule has a lot of torsion. So think about like, if you have a rubber band, right, and you twist it up a bunch of times, there's a lot of like tension within that rubber band, right? So you can think of DNA as a rubber band because it has all these coils in it. So there's a lot of like tension going on. So in order to stabilize that, what we do is we put this guy here. And his name is topoisomerase. This is another enzyme that this guy just sticks onto this closed end here and makes sure that this DNA isn't like all waving around like this. It just like keeps it stable again. So it's kind of working in conjunction with these SSB proteins here. Okay, so now we have the entire setup, and now we can actually start copying DNA. And just to give you some scope of what's going on, we call this. So when the DNA is opened up, we kind of have this open region, then we have these closed regions on both sides like this, right? So essentially what we have here is, this is what we call a replication fork, because it's like a fork in the road, right, in the DNA. And then we have another replication fork on this side as well. So essentially what we're going to do is we're going to focus on one side of this diagram here, but eventually we're going to like think about what's happening on both sides. So just know everything I'm describing on this side is actually happening on the other side at the same time, because you know the DNA needs to be opened both ways, and then it'll meet at the bottom like this. Okay, so and yeah, since it's going in both directions like this, we can call DNA replication in bacteria bidirectional, two-directional, bidirectional. Okay, let's continue. Before we move on to the actual copying process, let's do a quick review of how we define the direction that DNA strands are facing. So if you recall, we had something called 5' and 3', right? So we always referred to DNA in terms of the 5' end and the 3' end. So how that works is that if we go back to the structure of a nucleotide, and I'm not going to draw the entire thing, right? I'm just going to draw the basic structure, right? So we have here, we have our sugar, which in DNA is deoxyribose, right? So sugar, hopefully you can see that, yeah, sugar. Then we have the P, the phosphate group, right? Then we have our nitrogenous base. So let's say like A, for example, like adenine, it could be ATG or C. So here's our basic nucleotide. And basically we can number, so remember when I reviewed the chemistry part, we knew that each intersection of a line refers to a carbon atom, right? So essentially we have carbon, 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 and then, I should draw this out this way. Yeah, and then carbon. So if we go around, we can number these carbons. So we have, and we'll do this in orange. 1, 2, 3, 4, 5. And when we number these carbons, it's just convention to call them 1', 2', 3', 4', 5' here. So basically what we can do is that this sugar normally has what's called a hydroxyl group and an OH sticking off of it like this. So we can, basically when we look at DNA, we know that these nucleotides are going to be arranged in a long chain, right? As you see on the diagram. So here we'll have this, we'll have another one, we'll have another one, we'll have another one. You can see that at the end of each chain, right? Either at the top here, after you have billions of nucleotides, this P is going to be sticking off of the end here, whereas the OH is going to be sticking off of this end here. So basically this OH is on the 3' carbon of this nucleotide, right? So we call, after we have, a bunch, let's say these circles are nucleotides, we're gonna have this OH sticking off at the end. We're gonna call that the 3' end because it's on the 3' carbon, whereas here you have a bunch more nucleotides, and then we end with a P sticking off like this, and then that's on the 5' carbon, which means that this is the 5' end. So if we're looking at this long DNA strand, we can say that from top to bottom it goes 5' to 3' here. And then just a reminder that, so that's how we kind of orient ourselves to know which direction a DNA strand is like facing. But remember that if we have one strand going this way, the other strand is always anti-parallel, right? So remember this term, DNA is always anti-parallel. So essentially, if it's going from 5' to 3' on one strand, then the second strand, the complementary strand, is going to be flipped, where it's going from 3 to 5'. So essentially, what we're going to have is, if we simplify this diagram, we're going to have one strand is 5' to 3', then the other one is 3' to 5'. So you can see these two strands side by side, and remember, this has the P at the end, this has the OH, this has the P at the end, and then this has the OH. So hopefully that makes sense. Let me know if I need to explain that further, but I think this is a good segue into our next part, where we'll look at where the direction of the DNA will become very crucial in copying the DNA. So now we're looking at elongation. So we're going to start making that new DNA strand. Oh, and then another piece of terminology. This thing here that we're referring to, this is called the replication bubble, just because it looks like a bubble, right? Then as these guys are the DNA is getting split like this, the replication bubble is going to grow and grow and grow. Okay. Yeah, so we need to copy this DNA. So what we're going to need is a DNA polymerase enzyme. So DNA, I'm going to write it as DNA Pol. Every time I write DNA Pol, this means DNA polymerase, so an enzyme that polymerizes, creates DNA. And for we're going to use DNA polymerase 3. There's 5 types, I believe, of DNA polymerase, but this one is just the best for our purposes here. So DNA polymerase 3. This guy's going to copy our DNA, so he's our main character here. Now, the thing is that evolutionarily, some weird quirks have evolved with this specific protein. So number 1, as you can see on the slides here, number 1 is that this guy, he can't, so let's say you have a DNA strand here. Let's say this DNA Pol comes in. He can't just start making a strand just reading this other strand. So he's never going to be able to move along this strand on its own and then start making this complementary strand of DNA. That's never going to happen. The thing we need here is we need a short sequence here that's called a primer. So basically what this is saying is that DNA polymerase can't start building this new DNA on its own. It needs this starting point to be attached first in order to build off of this here. So it's like, think about Legos. This is my analogy that's in the slides. So like a Lego, like you can't just attach a Lego onto the ground, right? What you have to do is you have to put another Lego there first, and then use the Lego as a platform, and then you can start building stuff on top of that, right? So that's like the analogy that I came up with. I don't know if it makes sense, but essentially you just need this primer in front, and this primer is actually RNA. So the short RNA primer, it'll basically be inserted. So wherever you need to start copying, this enzyme called primase will come in and insert this RNA primer right here, the short sequence. Then the DNA polymerase can come up and then read and then start building off of that primer and then start making this new strand like this. So hopefully that diagram in the slides makes sense as well. Let's see if I can do anything else. Yes, okay, perfect. So there's kind of the first quirk of DNA polymerase 3. Now the second one is that this guy can only build DNA in one direction. So it can only, so it builds DNA off a free 3' end. So if we go back to that diagram we just looked at where we have the original DNA in white, we have that primer, right? We need, this primer needs to be 5' to 3' in order for the DNA polymerase to work here. So this DNA, right, DNA pol, can only recognize this primer if it's a free 3' end here. So it needs this 3' and then it can start building this DNA. So that means that if this needs to be 5' to 3', then the bottom, the original strand needs to be 3' to 5'. Right? So you can see this DNA polymerase can only build in one direction. So let's say we switch the direction of the DNA and we make it 3' to 5'. Oh, excuse me. I think that's literally what I just did. Let's make it 5' to 3'. So now this end is 3' and this end is 5'. So if we try to put a primer here, this DNA polymerase is not going to be able to build the strand because the free available primer has a 5' end instead of a 3' end. So this means that DNA polymerase essentially only can build DNA in one direction. And that'll be crucial in our next step here. So now we're going to zoom out once more, and we're going to look at our entire replication work. Here. This. So anything I draw in white is going to represent the old DNA, like the original cell DNA. So let's just assign arbitrary positions to these DNA strands. So let's say one of them goes from, I can't really see it here, so I'm just going to say this strand is 5' to 3', and then of course, if that one is, then the opposite strand needs to be 3' to 5'. So what we can do here is we can, so let's say we try to start, we're going to have to copy each of these two things. But the thing is, we can't, let's say we start here. Let's say we put a primer down here, then we try to transcribe the DNA in this direction. Now let's think about that. This primer is going to be 3' to 5', right? So we can't use this primer to build this way because this end here is a 5' end. So essentially, what we're going to do to synthesize or like make a copy of the top strand, what we're going to have to do is we're going to have to put a primer here. Now this primer will be 5' to 3', which means we have this available 3' end, and now DNA polymerase can come in, right, and then it's going to build, it's going to move this way, and it's going to build this new strand as it goes this way. Now let's think about how we're going to do the bottom strand. So if we want there to be a free 3' end for the DNA polymerase to build off of, we're going to need to put that primer right here. Because if the bottom strand is 3 to 5, then this primer is going to be 5 to 3'. So we have that available end, then our DNA polymerase can come in, DNA pol, remember DNA pol 3, and then it can start building the strand this way. So as you can see here, it looks like a pretty good system, right? We can put the primers in their appropriate locations, we can have the DNA polymerase move in the opposite direction, then we're going to copy all this DNA, right? Well, the problem, also, let me briefly mention that this DNA polymerase also is attached to what's called a sliding clamp. But we're not going to draw this from now on, but I'll just show you so that so that you've heard of it at least. So this, it will make sense if you look on the slides. The sliding clamp literally travels along with the DNA polymerase and makes sure that the DNA polymerase and the DNA are sticking together as they go, like this, instead of just falling off like that. So, another note. Yeah. So, all right, so this looks like a pretty good system, but there's a huge problem here, because remember, we have this DNA-B, this helicase here, constantly moving this way and unwinding the DNA, right? So what's going to happen is, right, as the helicase is opening this DNA up to the left side, we're going to get a bigger opening here, like this, as we go along, this thing here. So now we're opening the DNA even more. So now this helicase is all the way here. Now you can see, let's look at the top strand. So the top strand, there's not really a problem here because we had the primer here in the first place. So this DNA polymerase, you can just keep following the helicase and then keep transcribing and copying DNA as it goes all the way here, right? So no problem with the top strand. Now let's look at the bottom strand. The problem is that we placed our primer right here where we started originally, which means that this DNA polymerase is moving this way. So as this helicase is unzipping the DNA this way, we have this entire region here that's not going to get copied at all because we started the DNA polymerase here. So basically we need to solve this problem because we need all the DNA to be copied, right? So we want to look, how can we solve this problem? So what's going to happen is that, here, let me erase this so I have more space. This primase, right, the primase that put in, I'll just write as P, the primase that put in this RNA primer for the DNA polymerase to start with, what's going to do is it's going to have to jump back as the helicase is moving. This guy will jump here, and then I'll have to put another primer here. And now this DNA polymerase can come back and it can start making this strand this way, right? But you can see this as this process continues, we're unwinding the DNA further and further and further. And then this primase is going to continue having to jump. Now it's going to have to jump here and place a new primer. And now the DNA polymerase can go this way. So you can see as we go, this guy has to jump back, catch up, jump back, catch up, and it has to keep doing this process, whereas this top strand, the DNA polymerase can literally just keep moving in the same direction, this way. And remember, the entire reason for this is that DNA polymerase can only build DNA off of a free 3' end, and this primer provides that 3' end when it can only go this way, whereas the 3' end is here, so it can go this way from the top. So what we do is we call this top strand the leading strand. The leading strand. The top strand because it's leading the DNA replication process because it can just continue unbothered all the way through. Whereas this bottom one, we call it the lagging strand because it's kind of catching up to the leading strand because it has to keep, this primase has to keep jumping back, and then allowing this DNA polymerase to keep transcribing in the same direction, right? So here, here. So the second strand is kind of behind the other one. Yeah, so this is, okay, so now we've kind of come up with a solution to kind of transcribe all the DNA at the same time. Now the only thing here is that, remember, these primers are actually RNA. And we don't want our final product to have RNA in it because, the DNA is supposed to be, like, the genetic information should all be in DNA because DNA is the most stable, and we want to preserve the exact same DNA that we originally started with. Right, so what we're going to have to do is we're going to have to replace these RNA primers with DNA. So here, this is a very messy diagram, but bear with me here, so I'll erase some of this crap here. Oh, and let me talk about some terminology. So you see these like fragments of DNA that are kind of disjointed by these RNAs. These are called Okazaki fragments, named after the scientist that discovered how these work in replication. Okazaki fragments. So these bits of DNA on the lagging strand, these are the Okazaki fragments. So now we need to replace these RNA primers with DNA. So we have another player here by the name of DNA polymerase 1. So before we had DNA polymerase 3, that's moving along and making this DNA. Now we have DNA polymerase 1, which is a different kind of DNA polymerase. And what this guy will do is he'll come in to these regions of RNA primer, and he'll strip away that RNA. So now this RNA is stripped out, and this just gets degraded, just gets eaten up. And it'll come in and replace these regions with new DNA strands like this. Okay, so we solved part of our problem, right? So now we have one strand of DNA along the lagging strand. Now the problem is when this DNA polymerase removes, it takes the RNA out and it puts the DNA in. There's still these gaps between because it hasn't connected all the DNA together yet. So we still have this lagging strand with like a bunch of gaps in the DNA. So finally we're going to have to connect all these gaps, right? So what's going to happen is we get our final player here by the name of ligase. So ligase, this is another enzyme that'll come in, and it'll patch up these little gaps in the DNA, come in, just basically sticks a band-aid in all these broken regions here. Then it'll connect this into one continuous DNA strand. So that's how we solve the problem of the lagging strand here. And you can imagine as we open this up, we're going to have more Okazaki fragments keep being formed. Then all of these players, the DNA polymerase 1 is going to have to come in, replace the primers, ligase is going to stick them together again, and then you're going to keep moving, and then this process is just going to keep continuing all the way around the chromosome. So yes, perfect. And you'll see I have a overall diagram here that shows all the different enzymes within this pathway on the slides here. So let me know if I need to re-explain anything here, but hopefully that makes sense. And if we broaden our view a little bit, remember I said that DNA replication is bidirectional. So remember, we have two replication forks on each side around, and I'm going to draw, let's say the chromosome is this big circle, and then we have two replication forks moving this way. So then, remember we have, I'll just kind of simplify the diagram we had before. So we have leading strand, lagging strand, 3 primes. So this is just the exact same diagram before, so I just simplified it a little bit. The orange represents the RNA primers, then hopefully it's visible, yeah. And then the blue just represents the new DNA that's being formed. So we have the leading and lagging strands. Now if we go to the other side, it's going to be exactly the same, except now the leading and lagging strands are going to be switched. So we have this kind of middle point here, right? This is where we started the origin of replication where it split apart. Now on the other side, it's going to be the exact opposite. So this bottom is going to be the continuous, the leading strand. Then the top here is going to have these Okazaki fragments. So essentially on the other side of the replication bubble, the lagging and leading are going to be switched here. Then both will proceed as we go. And this makes sense because if we think about it, remember I said that DNA polymerase can only move this way, right? This arrow. So if we're on this side and we're trying to build this way, then we can't start here, we need a primer. We kind of have to do this process here, the same way we did here. Because here, right, the fork is opening up this way. As we go, we need to put a new primer where it's open, make the DNA copy, move new primer, make the DNA copy, new primer, etc., etc. So you can see on this side, this would be the lagging strand, whereas here, Now on the opposite side, even though this guy can go this way, now the fork is opening this way, so the DNA on what was before the lagging strand, the DNA polymerase can just keep moving this way and then continually go down this way. So this is a big picture of what we've been looking at, and these two things are happening at the same time, right? So finally, now that we've looked through that entire process, we can briefly look at termination. So obviously, so we have the replication bubble at the start, right? Have the replication bubble. As we go, this replication bubble is moving this way and this way, so it's getting bigger and bigger and bigger, right? Now the entire bubble is this thing. Then eventually you're going to get, once these replication forks reach the end, it's going to create this inner circle is just going to separate out from the new one, right? And then we have two chromosomes. So let me draw in the new DNA here. So here it's like this. This orange will represent the newly made DNA. So now you can see this outer ring has white on the outside, yellow or orange on the inside, like this. Whereas this inner ring, when it separates, it'll have the orange on the outside. So you can see our new two resulting chromosomes from replication each have half white, half orange. So half old DNA from the parent cell and half orange that was newly created. So this supports our idea of DNA replication being semi-conservative. Okay, that should be it. Just let me know if you have any questions and I can follow up if you guys need. But, yeah.